Welcome to today's Explainer. Today, we're looking at an absolutely massive bioengineering breakthrough coming out of KAUST. We're talking about nanoscale drug factories. This is a huge leap forward in programmable drug delivery, and it brings us so much closer to actually synthesizing therapeutics right at the site of a disease.
Okay, let's dive into this. What if your body manufactured its own medicine exactly where it hurts? I mean, imagine bypassing those awful systemic side effects completely. What if we could just hand your cells the blueprints and the tools to make the exact medicine you need right where you're sick? Well, as you'll see, that's the very real future this research is pointing toward.
The core premise here is just revolutionary. KAUST scientists have successfully engineered these tiny, nanoscale particles that literally enter living cells and transform them into miniature, highly specific drug-producing factories. We aren't just talking about delivering a pre-made drug anymore. We are delivering the actual biological machinery required to manufacture the drug right inside the human body.
Section 1. The Drug Delivery Challenge.
Now, you might be wondering, why hasn't this been done before? Well, it's because our cells have evolved incredibly strict security systems. Historically, just getting proteins inside is so tough that researchers usually only try to deliver one or two at a time. It is a huge struggle just keeping those few proteins alive and functioning. But the KAUST ambition? It was exponentially higher. They wanted to deliver a whole integrated multi-protein system all at once, acting as a single functional unit.
It was basically a cellular heist. Reich Grunberg, a senior research scientist at KAUST, actually called it a moonshot. He noted, "What we show here is that we can take a whole integrated protein system and bring it into human cells as one functional unit." They were attempting to smuggle an entire biological assembly line past the cell's rigid security systems, completely intact and ready to work.
Section 2. Building synthetic organelles.
To actually pull this off, the team used an incredibly clever material science trick: MOFs, or metal organic frameworks. Picture these as porous, sponge-like particles, engineered specifically to act as synthetic organelles. Essentially, they mimic the natural structures inside living cells. Try to visualize this MOF as a highly functional, protective bubble. The delicate proteins are packed safely inside this microscopic sponge, completely shielded from the harsh environment of the cell.
It wasn't easy right out of the gate, though. Initially, standard MOFs actually caused the proteins to lose their activity, so they had to be carefully redesigned. Naveen Kashab, a professor of chemical science at KAUST, explained it perfectly. She said, "By engineering a more porous, sponge-like framework, we were able to create an environment where the system could finally work as intended. By physically tweaking the holes in that sponge, they made sure the biological tools inside stayed perfectly active."
Section 3. A protein pathway transplant.
Now, what's really interesting about this slide is the sheer scale. 6. That is the number of proteins they successfully packaged and transported simultaneously. Delivering 6 proteins together as a coordinated system makes this the absolute most complex multi-protein system delivered into living cells to date. It's truly incredible.
So let's break down how this actually plays out inside the body. Step 1. The engineered MOF particles slip into the mammalian cells. Step 2. Thanks to that sponge-like armor we talked about, the cargo of 6 proteins remains totally active. And step 3. Acting like a tiny, intricate assembly line, they grab a simple amino acid already present in the cell and sequentially convert it right into medicine.
The specific medicine produced in this study is violacine, which is a bioactive compound being heavily studied right now for its therapeutic potential. The researchers are calling this a protein pathway transplant, meaning they didn't just deliver the medicine, they literally empowered the living cells to synthesize the therapeutic compound right on the spot.
Section 4. Programmable therapies and next steps. None of this happens in a vacuum. Stefan T. Arold, a professor of bioscience at KAUST, emphasized that combining expertise across biology and material science is really what unlocks these entirely new therapeutic approaches. I mean, this breakthrough simply wouldn't have happened without chemists, bioengineers, and material scientists breaking out of their usual silos and teaming up.
Now, to be responsible here, this work is still at an early proof-of-concept stage, but the road ahead is thrilling. The team is already planning to move toward animal model testing next. The ultimate payoff is immense. Because the platform is adjustable, scientists can actually program it, fine-tuning the protein interactions to target specific diseases. And the best part? By acting only at the exact site of the illness, this approach dramatically minimizes those unwanted side effects on healthy tissue.
So, are we entering an era where our own cells can be programmed to heal us from the inside out?
Thanks to these nanoscale drug factories, the answer looks a lot more like yes every single day. It is a profound shift from taking medicine to actually manufacturing it within our own cells. Thank you so much for joining me for this explainer, and definitely keep your eyes on this space.